Hi everyone and welcome back to the Bio Student Project. Today will be a continuation of the last video on glycolysis because today's video is about the other two steps of cellular respiration, the citric acid cycle and oxidative phosphorylation. First we're going to start with the citric acid cycle. This is the second out of 3 stages of cellular respiration and it goes by 3 different names: the citric acid cycle, the TCA cycle, or the Krebs cycle. They all refer to the same thing. This process takes place in the matrix of the mitochondria in eukaryotes, like humans, but in the cytoplasm of prokaryotes. There are 8 stages, and as it says in its name, it is a cycle. It occurs 2 times for each glucose molecule, so we produce a net 2 ATP. So now we are going to go over each of the 8 steps of the cycle. The enzymes used will be listed in the subscripts as usual. First, we start with acetyl-CoA, which we got after the oxidation of pyruvate, which is right after glycolysis, as we can see in this diagram here. The acetyl-CoA is joined with oxaloacetate, which we will see again at the end of the cycle, and citrate is formed. That is why it's called the citric acid cycle, because of citrate. Second, citrate becomes isocitrate, and they are isomers. To make this happen, a water molecule is both removed and then added. Third, isocitrate becomes alpha-ketoglutarate. A carbon dioxide molecule is released because, right now, these molecules have less carbon atoms than we started with, and NAD+ is reduced to NADH. Fourth is alpha-ketoglutarate, which joins coenzyme A to become succinyl-CoA. Another carbon dioxide molecule is released and NAD+ is reduced. Now we move to the 5th step. Even though it says 1 through 4 here, this is still the same process as the previous slide, so they are really steps 5 through 8. Succinyl-CoA becomes succinate after a phosphate group replaces the CoA, which then also changes GDP to GTP. GTP functions similar to ATP. It stands for guanine triphosphate, and it can also be used to generate ATP later. Succinate then becomes fumarate, and FAD becomes FADH2. These will come back later in the 3rd stage of respiration. Next, fumarate becomes malate with the addition of a water molecule, and finally malate becomes oxaloacetate with another NAD+ reduction. If you remember, we started step 1 with oxaloacetate joining acetyl-CoA. This makes sense because, again, this is a cycle, so one of the products is then used in the cycle again. So to recap the citric acid cycle, 2 carbons enter and 2 carbon dioxide molecules are released. We have 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of GTP or ATP. Again, this cycle occurs 2 times for the 2 glucose molecules, so these products will be doubled by the time the 2nd stage is finished. Now we are going to move on to the final stage of cellular respiration, known as oxidative phosphorylation. Oxidative phosphorylation is super important. As you can see from this diagram, you make around 34 of your ATP from just this stage, while from glycolysis and the Krebs cycle, you only make 2 each. Oxidative phosphorylation consists of 2 parts, the electron transport chain and chemiosmosis. Also, as you can probably tell from its name, oxidative phosphorylation needs oxygen. It occurs in the inner membrane of the mitochondria. In this process, ATP is synthesized using ATP synthase. You have protein complexes and cofactors, which we will go over in just a minute. Oxidative phosphorylation consists of a bunch of redox reactions. First, we need to orient ourselves within the mitochondria. We are looking at the inner membrane, which means around us is the intermembrane space, which is the area between the inner and outer membranes, as well as the matrix, which is the inside of the mitochondria. One important thing is that there is a very high concentration of protons in the intermembrane space compared to the matrix. You can see that here in this diagram. The protons naturally want to go to the matrix to balance out the concentration, but they can't fit through the membrane, so they have to go through ATP synthase, which is this purple area over here. The top of ATP synthase is like a motor. It spins when protons attach to it using something called the proton motor force. This allows protons to get to the matrix and powers ATP synthase to actually synthesize ATP, because it needs energy. The diffusion of the protons is known as chemiosmosis. So the second part, the electron transport chain, consists of complexes 1, 2, 3, and 4, as you can see here in this diagram. Complexes 1, 3, and 4 are proton pumps, which pump protons from the matrix into the intermembrane space. They do this because we need to maintain a high concentration of protons in the intermembrane space in order to keep ATP synthase spinning. So remember, there are cofactors on the complexes, which actually pass the electrons in the electron transport chain. The cofactors on complex 1 have a low electronegativity, and they get increasingly more electronegative as it goes to complex 4. This means that the cofactors are less greedy at complex 1 and the most greedy for electrons at complex 4. So, as the electrons are being passed down the chain, they are producing energy that is used for the proton pumps to actively pump protons back to the intermembrane space. Remember that I said that oxygen is needed for this stage. Well, oxygen acts as the terminal electron acceptor at the end of the chain, meaning it picks up the electrons after it's done with complex 4. It actually takes 2 electrons and 2 protons, making H2O, a water molecule. And with all of that, anywhere from 28 to 34 ATP molecules have been synthesized, and cellular respiration is all done.